Hello guys, and welcome to another episode of biology. And of course today we're looking at the internal structures of monocot and dicot roots, monocotyledonous and dicotyledonous roots. Okay, so do not forget that these these episodes are brought to you by the O3 Schools app. And of course, if you're preparing for your JAMB or your WAEC exam or any O-level exam at all, this is the right place to be, okay? And you are not the only one that is meant to be here alone. Invite all your friends or your loved ones, okay? Let them come and learn and pass at once. We'll talk about the app in a moment, but let us see a question. It's a JAMB from the JAMB app. This is a biology question 2000, year 2000, number 3. We got this using the question search feature on the app, all right? It has been displayed on that side of the board. It said, which of the following features are all associated with monocot? Okay, which of the following features are all associated with monocot? A says fibrous root system, branched network of veins, and one seed leaf. B says fibrous root system, 2 seed leaves, and floral parts in trees. C says 1 seed leaf, petals in 3 or groups of trees, and parallel venation of leaves. D says 1 seed leaf, net vein leaves and petals in 3s or multiples of 3s. Okay, so all of these are questions that you should expect in your exams. Okay, and that is why we're discussing all of them, and we'll try as much as possible to discuss it in details. Okay, so you do not miss any part. So please, we implore you, I'm advising you to download the O3 School JAMB app immediately, or the WAEC app, and start preparing for exam if you want to pass your exam at once. Okay, the app has all the past questions that you need for your exam, so you don't need to bother about getting past question anywhere. And what's more, there's also a classroom feature. Okay, we designed the classroom to meet a lot of your needs that you do not even know that you need. Okay, for example, many of you do not know that your JAMB syllabus is very important to your success in any exam, or your WAEC syllabus, and you must complete all. Okay, you must finish the full syllabus before you can say you are properly prepared for the exam. So the classroom has a study plan. The study plan has the JAMB syllabus, and of course, that study plan can create a schedule for you or a timetable that will make you easily finish your timetable. It has lecture notes, okay, that are structured according to the syllabus, right? That were designed to bring out the best in you. What's more, there's also the question search feature. Just like we will search for this question, okay? We just went to the app, clicked on question search. After clicking, we click on the subject biology, and then we just searched for dicot or monocot, okay? And the question that JAMB has set or WAEC has set under monocot or dicot just came out, voila, and then we just started answering, okay? So please, useful features. And there's also a UTME challenge or a JAMB mock challenge, okay, or the WAEC challenge also that hosts every Saturday. All right, so in that challenge, all students writing the exams for this year come to the app to compete with each other to win prizes every Saturday. The prizes is not the major point to, okay, the major point is for you to what, for you to get used to what, JAMB or WAEC past questions, to put yourself in place of an examiner, see how he's reasoning, how he wants to catch you, how he wants to set the question to confuse you. And of course, you see your result released every Saturday like that. All right, so please ensure you jump on the opportunity. Go to Play Store immediately or App Store, download the O3 Schools app, all right, and start preparing. Also, there's an activation fee, okay? It is on the app. It is enshrined on the app, okay? So please ensure you make your payment. Get your activation key. The price is less than the price of rice and chicken. Now, if you go to a restaurant and say you want chop rice and chicken, okay, the price is far, far lesser than that. So please, this is your future, okay? Rice and chicken, where you go shit come out for your bomb banana or ice cream. Okay, some of you buy custom ice cream of 8,000, 10,000, but subscribe for another app, it become a problem. So please, this is your future. Do not joke with it. So let us see the internal structures of monocot and dicot. First of all, what are monocot plants and what are dicots? Okay, and we already know that what monocots and dicots, they are the major types of what, of flowering plants, okay, that's known as what, angiosperms. Angiosperms, okay. So there are the 2 major types of what, of flowering plants, okay, that are known as what, as to call angiosperm. And of course, from our primary schools, we have been taught what monocots are or what dicot plants are, okay, and we know definitely that monocots, all right, as it was defined then, that monocots are what? Are plants that have what? One cotyledon. Mono means what? One. Then monocotyledon, all right? So they are called monocotyledonous what plants, okay? So they have one cotyledon or one seed leaf, okay? So monocots are plants that have what? One cotyledon, okay, or one seed leaf, okay? While dicots, okay, are plants that have what? Two cotyledon, dicotyledon, okay? So di means what? Two, means they have what? Two what? Cotyledon or two seed leaves, okay? So that is the simple definition of what? Of monocot and dicot. Monocots have one seed leaf, while dicots have what? Two seed leaves, simple as ABC. So now quickly we'll do something because it's very, very important. All right. I'm going to show you or write out the characteristics of monocots for you and the characteristics of dicots for you. Okay, so that you'll be well abreast of that. Questions come out from there from JAMB. So it's very necessary that what you will pay attention to it. Okay. So we're looking at internal structure of monocot and dicot roots. All right. And how do we get the internal structure? Okay, for us to get the internal, for us to be able to see the internal structure of a monocot or dicot root, okay, we need to take the transverse section. Okay, transverse. Okay, we need to take a transverse section that we call TS, transverse section, okay, of what? Of a monocot or a dicot root. Okay, and what does transverse means? It means cross-sectional what? Cross-sectional what area? Okay, we are taking a thin slice. We are cutting a thin slice along what? The perpendicular what? The longer perpendicular axis, okay, of the plant. And then what? Will now what? It will reveal to us the internal structures, okay, all of the tissue arrangement, okay, in the what? In the plant, all right? So now, before we talk about what this internal structure, let's quickly get ahead with what? With monocots and dicots, okay? So we said that what? Monocot, first of all, let us see monocots. All right. What do we know about monocots? Number one, we said that what? Monocots have what? One cotyledon. Okay. So they have what? One cotyledon. Okay. In terms of what? Cotyledon, they have what? One cotyledon. All right. So one cotyledon also means what? One what? One seed leaf. Okay. They have what? One single seed leaf. They have one single seed leaf. That is why we call them what? Monocot. Okay, one cotyledon or what? One what? Seed leaf. Also, we classify them in terms of leaf venation. Leaf venation. Okay, so if you look at what the leaf of what of a monocot, there is a way that what that leaves, okay, are what? Are arranged. Okay, so for monocots, they have what we call parallel leaf venation. Okay, they have what? Parallel what? Leaf what? Venation. Parallel venation. Okay, and of course, we can see this parallel venation in what? In some plants, okay, like grass. You can see them what? In grass, okay. You can see them in lilies. You can see them in lilies, okay, lilies. You can see them in maize, okay, maize or corn, all right? So these are what? These are characteristics of what? Of monocot. They have a parallel what? Leaf venation. A parallel leaf. You must take note of, in terms of leaf venation, they have, monocots have a parallel leaf venation. It's a very, very common question what? In JAMB or WAEC exams, okay? Also, classify them in terms of what? Flower parts. Flower parts, okay? How are their floral parts arranged? The flower part or the floral parts, how are they arranged? For monocots, they are arranged in groups, usually in what? In multiples of three, okay? Their floral parts are arranged in multiples of three. For example, we can have three, we can have 6, you can have what? 9, okay, and the likes, okay? So this is how their floral parts or their flower parts are arranged. Also, there are stem vascular bundles, okay? There are stem vascular bundles. For monocots, they are scattered throughout the stem, okay? We say they are scattered throughout the stem, okay? So for what? For a monocot, okay, the stem vascular bundle is scattered throughout what? The stem, okay? It's scattered throughout the stem. All right, so please take note of what of that. All right, also arrange them in terms of root system. root system. Okay, and for monocots, okay, monocots have what? A fibrous, take note. Monocots have a fibrous what? Root what? System. A fibrous root system. And what does this mean? It means that they do not have any main taproot. Okay, they have no main taproot. Okay, so monocots, their root system is what? It's fibrous. Okay, it's fibrous. They do not have any main what? Taproot. They do not have any main taproot. This is another what? Characteristic of what? Of monocot. Also, secondary growth is absent. Secondary growth, okay, in monocot is absent. Okay, is absent. All right, so they do not have what? Secondary what? Growth. They do not observe secondary what? Growth. Okay, like what we mean, like formation of bark. Okay, formation of what? Wood. All right, so they do not what? Have what? Secondary what? Growth. Okay, and of course, examples of monocots, like I mentioned here, some of them, like maize, You know what maize is called now? Agbado. Okay, one will call corn. Okay, that's what an example of what? Of a monocot. Okay, also we have rice, we have wheat, we have banana, we have lilies, we have onions. Okay, all of these are examples of what? Of monocot. Okay, maize, rice, wheat, banana, lilies, onions. All of them are examples of what? Of monocot. Please take note of what? Of these characteristics of what? Of monocot. Okay, so they have one cotyledon. They have a parallel leaf venation. Their floral parts are in multiples of 3. Okay, like 3, 6, 9, 12, like that. Stem vascular bundle is scattered throughout the stem. Okay, root system is fibrous root system. Take note, and secondary growth is absent for monocots. Okay, so now let us quickly dive to dicots. Okay, before we now move properly to what? To the internal structure of monocots and dicot roots. Okay, so now for dicots, we know that what? Dicotyledons. Okay, they have what? Plants that have what? 2 cotyledons. Di means 2. Okay, they have 2 seed leaves. Okay, so we know that for dicots, okay, we know that what? In terms of cotyledon, they have what? 2 cotyledon. Okay, or what? 2 seed leaves. Okay, please take note of that. All right, so they have what? 2 cotyledon. That is what? 2 seed leaves. Likewise, another characteristic quickly we'll look at is the leaf venation, as we looked at for the monocot. Leaf venation. For dicots, okay, they have a reticulate vein. Reticulate what? Vein. Okay, reticulate. By reticulate, we mean net-like what? Vein. Net-like what? Vein. Okay, so they have what? A reticulate what? Vein. All right, so dicots have reticulate vein. All right, but definitely, like we said for the monocot, they have a parallel what? Leaf what? Venation. Okay, this one they have reticulate what? Leaf venation. This it's arranged like a net. Okay, like a net. Like for example, we see those in what? In roses. Okay, in pea, in mango. Okay, they have what? Net-like. They have a reticulate what? Vein. Okay. Also, another classification is in terms of their flower parts or their floral parts. Their flower parts. And for dicots, their flower parts are arranged in 4 or in multiples of 4 or multiples of 5. Okay, so they are arranged in multiples of 4 or 5. Okay, so the flower part is usually arranged in multiples of 4 or 5 for dicots. Okay, now also the stem vascular bundle. The stem vascular bundle, this one for dicot is what? Arranged in a ring. It's not scattered like what? Like the monocot. Okay, so the stem vascular bundle is arranged in a ring. It's not scattered. It's not scattered like we saw for what? For the monocot. Now also their root system. Okay, for dicot they have what? A taproot system. Okay, so we say taproot what? System. Okay, they have one main root. They have one main root, and then they have many branches. Okay, so this is how what their root system what is, okay? They have a taproot system, okay? You have one main root system, then they have many branches, one main root, many branches, okay? Also, secondary growth for dicot, secondary growth, okay, is what is common, is present, all right? Like the formation of what? Of bark, okay, or wood, okay? Formation of wood, WOOD, okay? Or the one you call bark, okay? So it forms for that bark, okay? Wood is what? You can get wood from it, okay? So secondary growth is what? Is present, okay? And also, examples of plants, you can find this. We have listed was some rose, pea, mango, okay? Like you can see them in beans, okay? You can see them in tomatoes, okay? All right? You can also see them in sunflower, all right? All of these, beans, tomatoes, rose, pea, mango, all of these, they are what? Examples, okay? Of dicots. All right, so please take note of what this was characteristics. Okay, so for dicotyledons, they have two cotyledon or two seed leaf. Leaf venation is reticulate vein. Flower parts, usually multiples of 4 or 5. Stem vascular bundles, okay, arranged in a ring. Root system, they have a taproot system. One main root, several branches. Secondary growth is very common, okay? Unlike what? The monocot, that it is absent. So now let us now go to what's to the other part of this class. Because let me not say important, because everything we have discussed now is even very, very, very important. All right, so now let's go. But before we do this, please, I discovered that many of you do not support our efforts here. You do not even care. All right, many people are there failing exams, and it pains us that you are the reason why they are not aware of this channel. So please, want to implore you, want to beg you to please like this video. Even before you continue, just click on that like button that you are seeing, all right? Click on the like button, because liking the video will make it to be easily suggested to people who are looking for this kind of videos, all right? And also, ensure you subscribe to this channel. Subscription is free. It's just for you to get notified whenever we release a new update, a new video. All right. So also, why should you not click on the subscribe button? If you have subscribed before, no problem. All right. But if you have not, please click on that red subscribe button. Then likewise, ensure to drop comment if you have questions. We'll give you an assignment. Drop those assignments in the comment section. At the end of the class, they are usually what assignment. All right. So we'll drop them in the comment section. Okay. And do not forget to copy the link. Click on that share. Copy the link. Go and share it to your class group, to your mates. Let them come and learn. Let them come and enjoy learning and succeed. So now, the internal structure, like I said, can be gotten from what? From the transverse section. Okay, from the transverse section of a monocot or dicot root. Okay, so quickly want to take a look at what? At the dicot root. First of all, right, let's look at the dicot root. All right, we'll look at the structures, okay, the internal structures, okay, of a dicot root. They will now look at that of a monocot root, okay. So now, how does a dicot root even look like? Okay, so if you take a transverse section of a dicot root, okay, it looks basically like this. All right, it looks like this. I'm going to clean it. All right, something like this, okay? Then you now have 1, 2, 3. Don't mind my diagram, okay? All right, then here you have, okay? So this is how basically what it looks like. Okay, then it has what? Root hairs like this, root hairs like this, root hairs like this, okay? So this is basically how it looks like. Now, these ones are called the root hairs. Okay, I'm just going to show you some of the parts, okay? In case you want to take the diagram, you can just draw it, okay, in your book one kind, all right? Just for you to get an understanding of how it looks like. This is a dicot root. These are called what? This is a root hair. These are called root hairs, okay? So this is a root hair. Then here is the outermost layer, okay? The outermost layer is called the epidermis. Okay, it's called the epidermis. This epidermis is also called rhizodermis. Rhizodermis. Okay, you can hear it also being called the piliferous layer. Piliferous layer. All right. You can also hear it being called another name. Okay. Once I remember, I'll let you know. Okay, epidermis, rhizodermis, piliferous layer. It can also be called the epiblema. Okay, it's also called the epiblema. This is EO. Epiblema, all right? So these are the names you can call what the epidermis, all right? The outer layer, The outer layer. Then immediately after the epidermis, in here we have what? The cortex. All right, we have what? The cortex. Okay, and immediately after the cortex, the innermost part of the cortex, so this one now, is called the endodermis. Okay, it's called what? The endodermis. All right, then here definitely this round one inside here. Okay, this round one inside here is called what? The xylem. It's called the xylem. All right, then this one is called what? The phloem. Okay, this one is called the endodermis, while this inner one is called the pericycle. All right, so these are just some of the parts. All right, these are just some of the parts of what? Of a dicot root. Okay, we're going to take each of these parts one after the other. All right, one after the other, and see what their main purposes. Okay, and their functions in the dicot root. Okay, so now if I'm not too busy, let us begin with the dicot roots. Dicot roots. What are the features? Okay, what are the different parts that it reveals to us once we take that transverse section? Okay, look into the cross-sectional area. Okay, what does it reveal to us? So let's look at what? Dicot roots. Okay, so number one, the first part I'm going to look at is the epidermis. Okay, so the epidermis is also called the what? The epiblema. Okay, like I said, it's also called the rhizodermis. Rhizodermis. Okay, it's also called the piliferous layer. Okay, so now what does it reveal to us? This epidermis. What does it reveal to us? Number one, we know that the epidermis is the outermost layer, okay, of the cell. Outermost layer. So the epidermis is the outermost layer of the cell. Therefore, number one, we say it is the single outermost layer of the cell. Okay, it is the outermost single layer of cells. Okay, it is the outermost single layer of cells. That is what? The epidermis. Okay, and of course, aside this, it is thin-walled. The epidermis is thin-walled. Number two, okay, it is thin-walled without cortical. Okay, the epidermis is thin-walled without cortical. And of course, just like I was explaining to you, if you see, okay, the epidermis, this is the epidermis, and of course, this is the epidermis here. If you look at it you see on top of this epidermis, you can see some root hairs. Okay, so some of these root hairs, okay, come from what? This epidermis cell. Okay, so some of these root hairs come from the epidermis. So root hairs arise. Root hairs. These ones here are the root hairs. Root hair. So root hairs arise from some of these cells. Okay, please take note of that. And of course, number four, it has no stomata. The epidermis has no stomata or chloroplasts. So the epidermis cannot carry out photosynthesis. Okay, it has no stomata. It has no chloroplast. Okay, and then of course, most importantly, the epidermis is very important in the absorption of water and minerals. It helps in the absorption of water, okay, and minerals. Okay, so this whole you should know about the epidermis. Okay, so it is the outermost single layer of cells. It is thin-walled without cortical. Root hairs arise from some of these cells, and it has no stomata or chloroplasts. Okay, so it helps also in the absorption of water and minerals. Very, very important. Okay, and if you look at after the epidermis, the next part to look at is what? Is the cortex. Just after the epidermis, you see the cortex. Then before you now see the endodermis. Okay, so we're going to take that in that order. Okay, so let's look at the cortex. The cortex. All right, so please, like I said, please ensure you like this video, ensure you subscribe to our channel, share the link to your friends and loved ones, then they come and enjoy what they have missed. Okay, and do not forget to subscribe. So I was looking at dicot root, so we're looking at number 2 now, the cortex. Okay, and just like we said just now from the diagram, the cortex is located just below the the epidermis. Okay, so number one, it is located below the epidermis. Okay, and of course, this cortex, number two, is made up of what? Parenchymal cells. It is made up of parenchymal cells. And if you remember from the introduction to this was supporting tissue, we said that what? Parenchymal cells have large intercellular spaces. Okay, and so because this cortex has what parenchymatous cells, it means that what definitely it will also have a large intercellular spaces also within them. Okay, so number two, we say that it is made up of several, number two, made up of several parenchymatous cells, okay, with large intercellular spaces. Okay, with what? Large intercellular spaces. Okay, so that is that for that. So the cortex is made up of many parenchymal cells or parenchymatous cells. Okay, with large intercellular spaces. Now, also number three, the function now, it helps in the storage. Okay, helps in the storage of food, and also helps in the transport of water. Okay, please take note of that. So the cortex is located below the epidermis. It's located below the epidermis. It is made up of several parenchymatous cells with large intercellular spaces, and helps in the storage of food, and helps in the transport of water also. Okay, and remember very well that the innermost layer of the cortex is what we call the endodermis. Innermost layer. Okay, so you can add that that the innermost layer of the cortex is called the endodermis. The innermost layer of the cortex is called the endodermis. So now let us now go to the endodermis itself. Okay, let us talk about the endodermis. Okay, so the endodermis, like we have said, is the innermost layer of the cortex. Okay, and of course, number two, that's number three now. I'm talking about the endodermis. Endodermis, okay? And this endodermis is single layer of barrel-shaped cells. Okay, number one, it is a single layer of barrel-shaped cells. And what does barrel-shaped cell mean? Okay, barrel-shaped cell means that what? They are kind of cylindrical. They are cylindrical, okay, and rounded. Okay, at what? At both ends, just like this, just like something like this. Okay, then something like this. Okay, they are cylindrical and rounded at both ends. So the endodermis is a single layer of barrel-shaped cells. Okay, they are cylindrical and what? Rounded at both ends. Okay, and their cells have what we call Casparian strips. Okay, number two, their cells have Casparian strips. And by Casparian strips, what do we mean? We mean that what their cells have what we call a band. They have a band, B-A-N-D, band of suberin and lignin. Okay, they have a band of suberin and what and lignin that are located on radial and transverse walls. Okay, on radial and what? Transverse walls that helps what? It to what? To coordinate or conduct rather water and solutes. Okay, help to what? Conduct what? Water and solutes. Okay, so that is that for number two. They have what? Casparian what? Strips. Okay, they have bands of lignin and suberin. Okay, that helps in the conduction of what? Of water and solute. Now, number three, some thin cells, okay, that do not have Casparian strips, okay, they are called passage cells. Some thin cells cells, okay? Thin wall cells, some thin wall cells that do not have Casparian strips, okay? They are called passage cells. Passage what cells, okay? They are called passage cells. And what does this passage cells help them to do? It helps in the conduction of water, okay? It helps in the what? In the conduction of water, okay? So some of their cells have Casparian strips, but some do not have what? Casparian strips, okay? And those ones that do not have those Casparian strips, okay, they are called what? Passage cells. And then those passage cells help in the what? In the conduction of water, all right? So we say number 3, that some thin-walled cells, without, okay, without what? Casparian strips, okay, called passage cells. Okay, helps in the conduction of water. Okay, so this is the endodermis. We say it's a single layer of barrel-shaped cells. Okay, barrel-shaped cells. Put cells there. Single layer of barrel-shaped cells. Their cells have Casparian strips, and then some thin wall cells without these Casparian strips are the ones we call the passage cell. And what do? They simply help in the what? In the conduction of water. All right. So please take note of what? Of that. Okay. Why these Casparian strips? There are bands of what? Suberin and lignin located on the what? Radial and transverse walls that helps in the conduction of water and solutes. Helps in the conduction of water and solutes. Okay, so now that will take us to the next part, which is the pericle. So now let's look at the pericle. Let's look at the pericle. All right, the pericle. So number 4 now. Number 4. Pericle. So what is the pericle? Okay, like I showed you from the diagram. Okay, let me just quickly show you again. Okay, we now have this. Okay, this one is the epidermis. This one is the cortex. This one is the endodermis. All right. Then this one. All right. This other one here is the pericle. So it's just outside of the endodermis. This is the endodermis. This one here is the pericle. It's just outside of the endodermis. All right. So that is what the pericle is. The pericle. Now, so number 1, we know it's just outside. Number 1, it is a layer just outside the endodermis. Okay, so that's the pericle. And of course, the pericle is also made of parenchymatous or what? Sclerenchyma what cells. Okay, the pericle, it is made up of parenchyma or sclerenchyma what cells. Sclerenchyma. Okay, cells. Please take note of that. All right. So the pericle is made up of parenchyma or sclerenchyma cells. It's made up of parenchyma or sclerenchyma or cells. One will call the parenchymatous or the sclerenchymatous or cells. All right. That was what I was trying to pronounce. That I was speaking in tongues. Okay, so it's made up of parenchyma or sclerenchyma or cells. If you do not know what parenchyma is or sclerenchyma is by now, then that means you should not be in this class. Go and watch the first episode. We have done a great deal on all of that. Then number 3, it gives rise to lateral roots. Number 3, it gives rise to lateral roots. Okay, it gives rise to what? The lateral root and some parts of what? Of vascular cambium. Okay, it gives rise to lateral roots and some parts of vascular what? Cambium. Okay, which is very, very what? Necessary for secondary growth. Okay, which is necessary, which is important for what? For secondary growth. Please take note of that. Okay, so the pericle is very, very, very what? Important. Okay, now we say it is a layer just outside the endodermis. That one, it's a fact we have verified. Okay, that it's made up of parenchyma or sclerenchyma cells. We say it gives rise to what? Lateral roots and then some part of what vascular cambium, which is what important during what secondary what growth. Okay, now that is for the pericycle. So now that will take us to the next one, what we call the conjunctive what tissue. Okay, okay, let us look at before I talk about the conjunctive tissue, let us first of all look at the vascular bundle. Okay, let's look at the vascular bundle for the dicot root. Okay, vascular bundle. Vascular bundle number 5. Number 5. Vascular bundle. Vascular what bundle? All right. Now, first of all, for the vascular bundles, you know that we're talking about the xylem and the phloem, first of all, isn't it? Okay. And now the arrangement. Let's talk about their radial arrangement. Radial arrangement. Okay. You discover that the vascular bundle, xylem, the xylem and the phloem are arranged in alternate radii. Okay, they are arranged in alternate radii. Okay, so the xylem and the phloem, okay, of a dicot root, they are arranged in alternate radii. They are arranged in alternate radii. You must take note of that. And what does this mean? Okay, and what this means is that the xylem and the phloem, they are not arranged side by side. Side by side. They are not arranged, they are not arranged side by side. They are what? Arranged in alternate what radii. Just imagine it like the spokes. Spokes. You know that long thing now that's on the wheel now of a bicycle. Okay? They are arranged in alternate what radii. All right? So xylem is there conducting what water and minerals. Phloem is there conducting what food. So they are not placed side by side. They are arranged on what? On alternate radii. That's what we're talking about. So for the radii arrangement, okay, the xylem and the phloem, they are arranged in alternate what radii. They are not placed side by side. All right. So let us see number 1 for the vascular bundle radii arrangement. We say that what? Xylem and phloem are arranged in alternate radii. Okay, so that is that. For that, you must take note of what of that. All right. So we see that what that is xylem. The xylem. Let us talk about the xylem. See that what that is xylem. They are usually what in tetrach or pentac. Okay, in 4 or 5. Okay, in tetrach. Okay, tetrach or what pentac. Okay, or in pentac. Okay, they are arranged in what? In tetrach or in pentac. Tetrach means 4, pentac means what? 5. Okay, so they have 4 or 5 what patches. 4 or 5 patches sent there, and they are centrally placed. Centrally placed. Okay, so that's why if you look at what diagram again, okay, or something like this. Okay, this is what the xylem. Okay, then we now have what? Okay, we now have. All right. 1, 2, 3, 4. 1, 2. Sorry, my diagram is very, very small. Just take it like this. Okay. So this here is what is the xylem. The xylem, yeah. All right. Why for the phloem? You see they are arranged alternately. This one is the phloem. This one is the phloem. This one is the phloem. All right. They are not placed side by side. They are arranged what? Alternately. Okay. They are arranged in alternate radii. All right. This one said that the phloem. Okay, so please take note of that. So the xylem is centrally what placed, okay, in 4 or 5 what patches, as you have seen. All right, while what the and of course the xylem helps in what? In water conduction. Okay, water and what? And mineral conduction. Okay, water and mineral conduction. While what the phloem on the other hand, okay, the phloem on the other hand, okay, this one is helps in what? In the conduction of food. Conduction of what? Of food. Okay, all right. So it alternates. The phloem alternates with the xylem. It alternates with the xylem. Please take note of that. I hope you understand this very well. If you do not understand it, just ask a question in the comment. I'm sure that is very, very clear. But anyway, so I'll go over it one more time. Vascular bundles, the radial arrangement, the xylem and the phloem, like I said, they are arranged in what? In alternate what radii. All right. The xylem is what? Usually what? In 4 or 5 patches, in tetrach or pentac. All right. And they are centrally placed, as you saw from the diagram, at the middle, centrally placed. Okay. And helps in water and mineral conduction. The phloem on the other hand, okay, is what? it helps in what? In the conduction of what? Of food. Okay, and it alternates with what? With the xylem. It alternates with the xylem. So that is for the vascular bundles. So now let us talk about conjunctive tissue. This conjunctive tissue is very, very helpful also during secondary growth, okay? Formation of cambium during secondary growth, okay? Now, let us look at the conjunctive tissues, all right? Conjunctive tissue, that's what number? Number 6, right? Conjunctive tissues. All right, and of course, these conjunctive tissues, they are made up of parenchymal cells also. Okay, they are made up of parenchymal cells. So number 1, made up of parenchymal cells or tissues, and they are usually placed between what? The xylem and the phloem. Okay, so the conjunctive tissue is made up of parenchymal tissues, okay, and of course it is placed between what? The xylem and the phloem. All right, it's placed between the xylem and the phloem. That is the conjunctive tissue. Okay, like I said, it also helps. It plays a very, very important role during secondary growth. Okay, so what does this one do? Conjunctive tissue helps in food storage. Okay, number 2, it helps in food storage and gives rise okay to secondary it gives rise to what? To vascular cambium during secondary growth. Okay, very important. You must take note of that. All right. So the conjunctive tissues that are made up of parenchymal cells are usually between the xylem and the phloem. All right, and it helps in food storage and gives rise to vascular cambium. Cambium is very, very important. Okay, for thickening. All right, formation of that bark, formation of that wood. Okay, it's very, very important. So the cambium is very important. It gives rise to vascular cambium during secondary growth. Then let us now look at the pith. Pith, the pith, which is the last part we are looking at for the dicot root. Okay, so we're looking at what? The pith, Pith, and the pith of what? Of a dicot root. Take note, it's this one is an exam question. Okay, the pith of a dicot root is usually small or even absent. Okay. It is usually very small or absent, very small for dicot root. All right, take note. The pith is usually very, very small. While let me give you a tip. For monocot root, it's very, very large. It's very big. But for dicot root, it's very, very small or absent. Okay, it has parenchymal cells. All right, it has parenchymal cells, and it is usually centrally placed. So that's all about the pith. Okay, it is small or absent. Like I said, it is made up of what? Made up of parenchymal cells and located at the center. All right, so please take note of all of these characteristics of these internal structures, okay, of the dicot root that we have just looked at. Okay, and the funny thing is that the monocot root and the dicot root, their internal structures, their parts are almost the same. Okay, all right, just that there are some key differences, very, very key differences, and that is why we're looking at both of them interchangeably. All right, if not, if we're looking at the monocot now, you say, is it not the same one we saw for the dicot? No. Okay, at the end we'll do a summary, okay, or to talk about the differences between the monocot root and the dicot root. All right, so let us quickly look at the monocot root. Because of time, we'll be faster in terms of the monocot root. We have explained a lot when it comes to the dicot root. So for monocot root, so we have what's called the epidermis, the epiblema. Okay, the epiblema retains all its names. So okay, it's also called the piliferous layer. All right, it's also called what's it called? The epidermis. Okay, so now the epiblema, it is the outermost single layer of cells. So I'm going to be fast, okay, because they have a lot of similarities, but definitely we're going to point out the differences at the end of the class. That will be the end of the class. So outermost single layer of cells. Okay, so what's the second one? The epidermis. We also know that what the epidermis, what is thin wall. So for monocot roots, okay, same as dicot root, the epidermis or the epiblema is thin walled. Okay, and what other thing was there? It has no cortical also. Okay, it is thin walled. Okay, has no cortical, and of course, also for the what's it called? For the monocot root, all right, you see some root hairs on the epidermis also. So some of these root hairs also arise from what? From these cells. Okay, so some root hairs arise from what? From cells. All right, and then lastly, it has no stomata also. Okay, the epidermis of a monocot root has no stomata. But we are trying to be very fast with this. Okay, has no stomata or chloroplast. All right, so these are all of the characteristics of the epiblema of a monocot root. It is exactly the same as we saw for the dicot root. All right, so now let us see. Let us now go to the next part. After the epiblema, what is the next part? We'll now see the cortex. Okay, so for the cortex, all right, we say it has several layers of what? Of parenchymatous cells. Okay, we say number one, it has several layers of parenchymatous cells. Okay, that means it has several layers of what? Of parenchyma cells. Simply put, okay, it has several layers of parenchyma cells. Then number two, cells have, of course, because they have what? These parenchyma cells, not generally or naturally, parenchyma cells have what? Intercellular spaces between them. Okay, so you can say that what cells have intercellular spaces. Okay, I hope I'm not too fast. All right, so it's just almost close to what I've talked about for the dicot root. All right, then number three, functions in storage of what? Storage of food and water transport. Okay, number three, storage of food and helps in what? In water transport. Okay, so that is that for the cortex of a monocot root. All right, please just write them down compared to that of the dicot. Okay, and you see that they're almost what the same. Then the ones that have differences, we will know very soon, okay? Because we are going to look at the differences together. So immediately after the cortex, what do we get to? Get to the endodermis, right? So now for endodermis, okay? That is the innermost part, okay, of what? Of the cortex, all right? So it is the innermost layer of the cortex, number one. It is the innermost layer of cortex, all right? And what do we remember from the last class? We said it is what? Barrel shaped, okay? It is barrel was shaped, all right? So they have what? The endodermis have compactly arranged cells in the monocot, okay? They are barrel shaped, number two. By barrel shaped, mean that they are cylindrical and rounded at both ends, all right? So we say barrel shaped. Barrel looks like a barrel. You know what a barrel is now, okay? It looks like a barrel. Barrel was shaped. Okay, and then they have what? Compactly arranged cells. Okay, then of course, number three, we talk about the presence of Casparian strips, isn't it? So also Casparian was strips. Casparian strips, okay, are what? Are present. Okay, are present. Casparian strips are present. Okay, but this time they are not present on both radial and transverse walls. Okay, they are only present in what? On radial walls. Casparian strips are present on radial walls. Okay, for the monocot root. Remember, for the dicot root, we said Casparian strips are present on both radial and transverse walls and helps in what? Conduction of what? Of water and solute, isn't it? Now, for the endodermis of a monocot root, Casparian strips are present based on what? On radial walls only. And then also we talk about passage cells, isn't it? All right. So passage cells are also here, IV now. So they also have passage cells, okay? Passage cells. These passage cells, they contain passage cells. These passage cells, they are thin walls, okay? They are thin wall, okay, and then what they do is that they allow water to enter the steel, and then they allow water to enter the steel. Okay, please take note of what of all of these what characteristics, okay, of the endodermis. The innermost layer of the cortex, barrel-shaped, compactly arranged cells, Casparian strips are present, all right, on radial walls, and then passage cells, thin wall, allow water to enter into the steel. This steel is the central part of the root. Okay, please take note. This steel, central, is the central part of the root. Okay, so what the passage cells do is to help what allow water to enter into the steel. Okay, very quiet, profound. Now, after the endodermis, that takes us to the what? To the pericycle. Remember? Yes, so we now have what? The pericycle. Just outside, located just outside of what? Of the endodermis. We have the pericycle. So a single layer of thin-walled parenchymatous or cell. That is the pericycle. We're talking about the pericycle. All right. We see number one. It is a single layer of parenchymatous cells just outside the endodermis. Okay, it is a single layer of parenchymatous cells present just outside the what? The endodermis. That is the pericycle. All right. Should I draw it? I think we have seen pericycle several times. So it's just almost the same what location with the word that of the dicot. Okay, and then gives rise to lateral roots. The pericycle gives rise to lateral roots. All right, and then of course in monocots, we saw that the pericycle is very useful during what secondary growth. Sorry, in dicots rather, we saw that the pericycle is very useful during what secondary growth. That is the formation of vascular cambium. The formation of vascular cambium in the dicot root. But for the monocot root, it gives out to lateral root, okay, but it does not form vascular cambium, because vascular cambium is absent in the monocot root, right? So take note of that. So it gives rise to lateral root, and then in monocots, number 3, in monocots, it does not give rise to vascular cambium. Okay, so that is why secondary growth is absent in monocot root. Please take note of that. All right, so now let us now go to the next one. And immediately after the pericycle, we have the vascular bundles. Okay, so let us look at the vascular bundles. All right, vascular bundles for the monocot roots. Vascular bundles. All right, so I have vascular bundles. All right, and of course, we're going to also talk about the xylem and the phloem, unfortunately, because those are the vascular bundles that we have. So in terms of arrangement, okay, we are radial, okay, they are radially arranged, okay. So the xylem and the phloem also of a monocot root, they are not placed side by side, okay, they are placed alternate with each other, okay. So then also they are polyarch. Okay, polyarch. The strands, okay, of xylem, okay, and phloem, or xylem and phloem bundles are usually many when it comes to monocot root. Okay, they are polyarch. They are usually either 6 or more. Okay, they are usually 6 or more. They are polyarch. So we say they are radial and polyarch. Okay, the xylem and phloem, they are arranged, they are alternate to each other, okay, in the alternate radii, while they are polyarch, they are bands, okay, their bands are more than 6, okay, 6 or more usually. All right, so now the xylem, first of all, as you see, the xylem, okay, the xylem alternates, okay, with the phloem, okay? The xylem alternates with the phloem, while the phloem the phloem lies between the xylem bundle. The phloem lies between the xylem bundle. Please take note of that, all right? So the vascular bundles, they say they are radial and polyarc, okay? By radial means they are not arranged side by side. They are arranged all in the alternate radii, all right? And we say that the xylem alternates with the phloem, while the phloem lies between the xylem bundle, okay? And this will now take us to the next part, which is what? The conjunctive or tissue. So let's talk about the conjunctive tissue, just like we saw in the last, in Dicot root, okay? Also, the conjunctive tissue, they are made up of parenchymatous or cells, okay? So, conjunctive tissues, okay? So, parenchymatous, number one, parenchymatous cells that lie between the what? The xylem and the phloem. All right, so take note of that. All right, and of course, they do not become meristematic, and so they do. That is why I say that our monocot do not have a secondary growth. Okay, they do not become meristematic, so that is why they do not usually would have secondary growth. So number two, they do not become meristematic, hence no secondary growth. All right, and then lastly, that will now take us to the pith, P-I or T-H. Okay, we know the pith, isn't it? All right, so for the dicot root, to say the pith is what? Small or absent. But for the monocot root, it is what? Well developed. Okay, the pith is well developed. Okay, and large, very big, but no good. Okay, it's well developed and large, and of course it's made up of parenchyma cells also, like we said for the dicot root. Okay, made up of parenchyma cells or parenchymatous cells. Okay, made up of parenchymatous cells, and also the pith is very important for storage. Please take note of that. So the pith of the monocot root and the dicot root, okay, you are seeing differences. So for the pith of the monocot root, it is well developed and large, while the pith of the dicot root, it is what? Small or absent. So now let us now go to what? To differences between what? What is it called? Differences between monocot roots and dicot roots. Differences between monocot roots and dicot root, and we go to answer questions and we're done. Okay, so differences between monocot root and dicot root. So this is a monocot, okay, and then this is what? Dicot. All right, so number one of the very common differences we have noticed, okay, from our class. Number one is that the monocot has a very well developed pith. Okay, so number one, I can say the pith, it's well developed and what? And big or large and large. Okay, so the pith is well developed and large. Okay, but for the dicot, okay, you know that what? The pith is what? Is small or absent. Okay, so we know that what? They both have what? Parenchyma or cells in them. The pith in the dicot and the pith in monocot both have what? Parenchyma cells in them. Take note of that. All right, so now the monocot has higher number. It is polyarc. Okay, it has higher number of what? Of xylem and what? And phloem. Okay, while the dicot is what? Is tetrarch. Okay, it's tetrarch. All right, so you can say number two has higher number of xylem and phloem bundles. Okay, this one has what? Has limited. Okay, has limited number. Has limited number of what? Xylem and phloem bundles. Okay, so also the pericycle for what? For the monocot, okay, it gives rise to lateral roots only. Okay, the pericycle for the monocot root gives rise to what? Lateral root only. Why this one gives rise to what? To vascular what? Cambium. Okay, that's why its secondary growth is what? Is present in what? In the dicot. All right, so number three, we say number three, okay, gives rise to lateral roots only. All right. While what the pith, while what the pericycle for the monocot root, okay, gives rise to cork cambium, okay, also gives rise to part of vascular cambium and lateral roots. Okay, hope you are seeing the difference is very clear. Okay, so the monocot root gives rise to what? Lateral roots only. All right, while what the dicot, okay, gives rise to what? Cork cambium, gives rise to part of vascular cambium, and of course lateral roots. Also, number 4, the xylem is round or oval. Number 4, the xylem is round or oval. Okay, for the what? For the monocot root. Why what? The xylem here, the xylem is angular or polyangular, as you may have it. Okay, please take note of that. All right. So the xylem here, the shape is round or oval, while the xylem here is angular or polyangular. Likewise, secondary growth we have seen here is absent. Secondary growth is absent. Okay, why here? Secondary growth is present. All right. And of course, for the monocot root, it does not have what? Cambium. Okay, cambium is absent. All right, so you can say cis cambium is absent. Okay, yeah, cambium is present. Okay, for the what? For the dicot root. Okay, then also the cortex. The cortex for the monocot root is very wide. Okay, the cortex for the monocot root is very, very wide. While the cortex for the dicot is narrow. Okay, the cortex here is what? Comparably what? Narrow. Comparably narrow. All right, please take note of what? Of that. All right, and then of course I've seen that what? That the xylem is what? Is polyak. Okay, while what? The dicot is what? Is tetrach. Okay, so you can say this one is polyak, has many what? Xylem and phloem bundles. This one is what? Tetrach. All right, so that is that for that. So these are the differences we have been able to draw between the monocot and the dicot root. The pith is well developed and large. Pith is small or absent. It has higher number of xylem and phloem bundles. This one has limited number of xylem and phloem bundles. Gives rise to lateral roots only. This one does gives rise to both cork cambium, part of vascular cambium, and lateral roots. The xylem is round or oval here, while the xylem here is angular or polyangular. Secondary growth is absent here, secondary growth is present. Cambium is absent, cambium is what? Is present. The cortex is very wide, while the cortex is comparably very narrow. So now, let us go to the JAMB app, O3 Schools JAMB app, answer questions now. We will try as much as possible not to waste so much time on them. This is a 2000, year 2000, number 3, year 2000, number 3. So which of the following features are all associated with monocots? Okay, I will not answer this question. You answer it and tell me the answer in the comment section. Okay, year 2000, number 3. In fact, let me just give you as an assignment. Year 2000, number 3. All right, then let us go to the next question. 2004, number 11. 2004, number 11. We say the internal structure of plants, a wide pith in the center is common to what? Is common to, let's say, dicot stems and monocot stems. All right, we have not talked about dicot stem. We'll talk about dicot stem, okay, in the next class. Okay, dicot and monocot stem in the next class. Okay, so, but it's not actually what? Dicot stems and monocot stems. All right, it's one of them also has a wide pith for the stem. Okay, but for the root, you know, the one that has what? A wide pith. Okay, so I will not answer this question also. Go to the app, look for 2004, number 11, the O3 School JAMB. Download it. Okay, activate it and start using it now. All right, you want your success. Okay, jump on it. Okay, so B say dicot stems and monocot root. C say dicot root and monocot root. D say dicot root and monocot stems. All right, Then now let us go to 2014 number 13. Okay, as you can see from the diagram, they say use the following to answer the question that follows. They say the diagram is the transverse section of a say dicotyledonous stem, B say monocotyledonous root, C say dicotyledonous root, D says monocotyledonous what? Root. Okay, and obviously there it is what? It is a dicotyledonous root, option C. Now, and how do you know? Okay, it has what is central what shaped what xylem. That's what is xylem at the what at the center. All right. Then also phloem between what xylem what arms. All right. And then of course it does not have a central pith. Okay, those are what the characteristics of what of a dicotyledonous root. Then now let us see 1994 number 12. Say use the diagram below to answer the question below. They say you are given the transverse section of a dicotyledonous root. Okay, they say starch is usually stored in the part labeled A says I, B says I, C says I, D say IV. Where will starch? Where is food stored? Where is food stored? Okay, so please, I want you to give me the correct answer. All right, right. So now, correct answer. That's year 1994 number 12. Okay, so 3 assignment: 2004 number 11. 2000 number 3, 1994 number 12. All right. So let me get the answers in the comment section. Okay. Thank you for watching. I'll spend a lot of time. I'll see you in the next episode. We'll talk about dicot stem and monocot stem.